Erythrocyte metabolism, and membrane structure and function. So there are phases of glucose catabolism in hematology. So red blood cells rely exclusively on glucose metabolism for energy since they don't have a mitochondria. Therefore, glucose catabolism in red blood cells occur mainly through glycolysis and related pathways. The first phase is the anaerobic phase, which is called glycolysis. The location of this occurs in the cytoplasm of the red blood cells. So glucose is broken down to pyruvate via glycolysis. The red blood cells convert pyruvate to lactate because they don't have a mitochondria. So key points to remember is it produces 2 ATP per glucose. This is critical for red blood cell energy needs. There is no Krebs cycle or oxidative phosphorylation in the red blood cells. NAD+ is regenerated by converting pyruvate to lactate. To remember importance is it provides ATP needed to maintain red blood cell membrane integrity in ion gradients. The second phase is a pentose phosphate pathway, or the alternative pathway. This occurs in the cytoplasm, and it parallels to glycolysis. So the pentose phosphate pathway, generates NADPH and ribose 5-phosphate. So NADPH is critical for maintaining reduced glutathione. It protects red blood cells from oxidative damage. Ribose 5-phosphate is used for nucleotide synthesis. So the importance of this step is it protects red blood cells from oxidative stress and maintains cell viability. The third phase is not really applicable to red blood cells because the red blood cells lack the mitochondria, so there's no Krebs cycle that occurs. But the energy generation is limited to glycolysis and the PPP. Enzyme deficiencies that are relevant to hematology is we have certain enzyme deficiencies that can affect glucose metabolism in red blood cells, can actually cause hemolytic anemia or other hematologic disorders. We'll talk about pyruvate kinase deficiency. This is the most common glycolytic enzyme deficiency in red blood cells. It results in decreased ATP production, which results in an impaired membrane ion pump, which causes hemolysis. Symptoms of that would be chronic hemolytic anemia, jaundice, and splenomegaly. We also have glucose 6-phosphate dehydrogenase deficiency. This is an enzyme in the pentose phosphate pathway. Its deficiency leads to a decreased NADPH, which then goes to a reduced glutathione depletion, which goes to an increased susceptibility to oxidative damage. This is triggered by infections, certain drugs, or food, such as fava beans. Symptoms of this is they could have episodic hemolytic anemia and dark urine. Next one is hexokinase deficiency. This is rare, but it does impair the first step of glycolysis. It leads to a decreased ATP production, which can lead to hemolytic anemia. There are a couple other less common ones. We have phosphofructokinase deficiency and also aldolase A deficiency. If you're in my class, there is a video there that describes the phases of glucose catabolism. It's red blood cell morphology and volume. So red blood cells, as mentioned before, have a biconcave disc shape, which helps to optimize surface area for gas exchange. The average red blood cell volume is about 90 femtoliters. This shape also contributes to flexibility and efficient flow through microvasculature. For red blood cell deformability, so red blood cells can stretch up to 2.5 times their resting diameter size without any damage. This ability allows them to squeeze through narrow capillaries and splenic pores. The deformability can depend on 2 main factors, so geometry and viscosity. Geometry, because of the biconcave shape, it reduces thickness and can increase the surface area. And the viscosity, so it has the cytoplasmic viscosity, which is mainly just a hemoglobin content, and it has membrane flexibility. So the importance of deformability is it's essential for red blood cells so they can navigate the microcirculation and splenic filtration. It also enables efficient oxygen delivery even through very small vessels. So loss of deformability and red blood cell clearance. So as these red blood cells age, they will lose membrane flexibility and deformability. So this basically results in the older red blood cells not being able to pass through splenic pores efficiently. So these senescent or older red blood cells become trapped and are phagocytized by the splenic macrophages. This is a mechanism for red blood cell turnover and maintenance of your healthy blood. Clinical relevance, so disorders affecting red blood cell membrane proteins or cytoskeleton, like hereditary spherocytosis, can reduce deformability, which can cause premature red blood cell destruction and hemolytic anemia. There is also altered deformability, which can also affect blood flow and oxygen delivery. The red blood cell membrane lipids. So there is a lipid bilayer structure of a red blood cell membrane. So the red blood cell membrane is a fluid lipid bilayer that provides structural integrity, flexibility, and selective permeability, which is essential for the red blood cells to function. So the composition of the lipid bilayer is, first thing, cholesterol. Makes up about 25 to 50% of the membrane lipid. The function is to modulate membrane fluidity and stability. It prevents membranes from becoming too rigid or too permeable, and it inserts between phospholipid tails, which helps fill the gaps. Next is phospholipids. This is a major lipid class, which is arranged in two layers with hydrophobic tails inward and hydrophilic heads outward. Main phospholipids will include phosphatidylcholine and phosphatidylethylamine. My goodness. The functions is it provides membrane barrier, participate in cell signaling, and influences membrane curvature and deformability. Glycolipids are another part of the bilayer. This is located primarily on the outer leaflet. Its function is it's involved in cell recognition and protection, contributes to the negative surface charge of the red blood cells, which is important for the prevention of clumping. So we have hydrophilic and hydrophobic structures. So hydrophilic heads of the phospholipids and glycolipids face the aqueous environment. Hydrophobic fatty acid tails face inward, which create a nonpolar core that restricts passage of water-soluble substances. The membrane asymmetry, so the lipid bilayer exhibits asymmetry in phospholipid distribution between the two leaflets. So the outer leaflet is rich in that phosphatidylcholine in sphingolimion, where that inner leaflet is rich in phosphatidylethylamine and phosphatidyldyslorine. This asymmetry is actively maintained by enzymes. The physiological importance is it keeps the phosphatidylserine inside, and exposure on the outer leaflet signals apoptosis or red blood cell removal. This also maintains membrane stability and cell recognition. Some more red blood cell membrane proteins. So they have integral proteins that span the entire lipid bilayer and are embedded firmly within the red blood cell membrane. They play crucial roles in transport, adhesion, signaling, and blood group antigen presentation. So the functions of the transmembrane proteins are transport sites. They facilitate movement of ions, metabolites, and gases across the red blood cell membrane. And they're also essential for maintaining ion gradients and cell volume. And then you have adhesion sites. These mediate interactions between red blood cells and other cells or the extracellular matrix. It's important for vascular health and immune recognition. And then we have signaling receptors. This detects extracellular signals and transmits them inside the cell. It helps regulate red blood cell responses to any environmental changes. Then we have red blood cell group antigens. So many blood group antigens are proteins or glycoproteins located on these integral proteins. Critical for blood transfusion compatibility. So if you haven't taken blood bank yet, you'll know all about that in a while. Some structural characteristics. Integral proteins are part of a large membrane macromolecule complex. These complexes function as transporters, structural components, enzymes, receptors, and adhesion molecules. The system involves 2 transmembrane lipoproteins and 1 multipass glycoprotein. Each of these proteins crosses the membrane 12 times, forming complex structures which are needed for red blood cell function. The cytoskeleton, so these proteins are peripheral membrane proteins located on the inner or cytoplasmic surface of the red blood cell membrane. They form a cytoskeletal network that supports the lipid bilayer and maintains red blood cell shape and flexibility. So for structure and proteins, we have spectrin, which is a main structural protein that forms a flexible lattice-like network. It's composed of alpha and beta subunits, and it provides mechanical stability and elasticity to the red blood cell membrane. Then we have ankyrin, and that links spectrin to the transmembrane protein band 3, and it acts as a bridge between the cytoskeleton and lipid bilayer. Then we have protein 4.1. It stabilizes the interaction between spectrin and actin filaments, and it enhances the membrane cytoskeleton association. Then we have actin and tropomyosin. Short actin filaments form junctional complexes within the cytoskeleton. Tropomyosin stabilizes actin filaments. Then we have adenosine and tropomodulin. Adenosine caps actin filaments and promotes spectrin-actin association. Tropomodulin caps the pointed ends of actin filaments, regulating filament length. And last one is we have demetin, and it is an actin-binding protein contributing to cytoskeletal stability. This is important for maintaining membrane flexibility. The function is a lateral or horizontal membrane stability. These proteins provide horizontal stability, maintaining the biconcave shape and flexibility of red blood cells. They also help stabilize spectrin, preventing membrane deformation under shear stress. And the cytoskeleton allows red blood cells to deform as they squeeze through narrow capillaries, but they can return back to their original shape and size. Any disruption of these proteins can lead to hereditary hemolytic anemias, like hereditary spherocytosis, due to a membrane instability. So membrane deformations, as we kind of mentioned, so red blood cell membranes deformation is critical for red blood cells to pass through narrow capillaries. Deformation relies on the mechanical properties of the red blood cell membrane, primarily governed by that membrane protein and their interactions. Mechanism of membrane deformation in the red blood cells. So the red blood cell membrane is a lipid bilayer, which is supported by an underlying cytoskeletal network. Membrane proteins connect the lipid bilayer to the cytoskeleton, which helps enable flexibility and mechanical strength. Key processes in membrane deformations, we have elastic deformation, which is a reversible change in shape, and mechanical stability, which is the resistance to rupture or cause permanent damage. So flexible interactions, so the cytoskeletal network mainly consists of spectrin tetrameres, actin filaments, and associated proteins. Major membrane proteins involved are spectrin, ankyrin, band 3 protein, protein 4.1, and protein 4.2. So these proteins help maintain the membrane's elasticity and mechanical resilience by flexible dynamic interactions. Any genetic mutations that can affect the red blood cell membrane protein and deformation. So mutations in genes coding for membrane or cytoskeletal proteins can disrupt the flexible, normal of the red blood cell. This can result in altered membrane elasticity, reduced mechanical stability, and abnormal red blood cell shapes. So first one up is we have hereditary elliptocytosis. This is caused by mutations primarily in the alpha or beta spectrin or protein 4.1. It affects, so the disruption of spectrin tetramere formation will cause a weakened cytoskeletal network. This causes the red blood cells to become elliptical or oval shaped, so we call them elliptocytes. And this causes reduced deformability, which can lead to hemolysis and anemia. And then we have hereditary spherocytosis. This is caused by mutations in ankyrin, band 3, spectrin, or protein 4.2. This is the effects of this is it can have a loss of membrane surface area due to defective linkage between cytoskeleton and lipid bilayer. So this will cause a formation of spherical red blood cells or spherocytes with decreased elasticity, and this can cause an increased fragility, which can lead to hemolytic anemia. And then we have osmotic balance and permeability. So definition is we have a selective permeability, which refers to the ability of the cell membrane to regulate the movement of substances into and out of the cell. This property allows the cell to maintain internal homeostasis by controlling the internal environment. Substances that can pass through freely through the membrane. So there are certain small or uncharged molecules that can diffuse freely across the membrane, and that includes water, bicarbonate, and chloride. So these molecules can move passively following their concentration gradients. So role of that cation pump, so sodium potassium pump is actively regulate the concentration of ions such as sodium, potassium, and calcium inside the cell. These pumps help to maintain ion gradients, which are critical for your cell volume regulation, osmotic balance, and membrane potential. So if there is a damaged cation pump, you can have some issues. It can cause an excess of calcium and sodium to accumulate inside the cell. This will disrupt osmotic balance and cause a water influx, which will lead to the cell swelling. Swelling can impair cell function or can actually lead to cell lysis. So role of calcium in red blood cell osmotic balance. Calcium plays a role in maintaining osmotic balance. The proper regulation of intracellular calcium levels prevents excessive water influx and preserves red blood cell shape and flexibility. So dysregulation calcium levels contribute to membrane instability and impaired red blood cell deformability.